Okay. So last time, I put all the stuff on Blackboard so I can pull the files up there, fortunately. But last time we had gotten getting to about this point right here. So we have transcription initiation. We have translation over here. And hopefully in general biology 1 you learned about RNA processing, which at this point I don't think we're going to get to today. So we had gotten to initiation. And then this is all elongation. And then we actually have transcription termination right here. And we talked about basically the protein factors that are necessary for initiation to occur. And we were talking about this in the context of bacteria, right? Because it's very easy to study that system. Bacterial RNA polymerase is, I think we talked to as many as 6 proteins. Alpha, alpha, beta, beta prime, sigma, and omega. And we talked about sigma has a puzzle piece shape that clicks very, very tightly into a specific DNA sequence. That DNA sequence was defined by David Pribnow. So sigma binds very, very tightly, which causes a problem in that so it binds in 2 different spots. When the polymerase wants to start transcribing down, it is locked into place. It's held into place by sigma. And the solution is to drop sigma behind, and the core enzyme goes down instead of the holo enzyme. And the energy that causes that release, that causes the alpha, alpha, beta, beta prime to release sigma, is when it's adding nucleotides on, and those ATPs, essentially those phosphates, are coming off. There are numerous, what we defined as abortive transcripts. Is that thing on right now? Okay, great. There are numerous abortive transcripts that are produced before it eventually goes off. The instant, the molecular event that defines, okay, this is important, you might want to write this down. The molecular event that defines transcription initiation is release of sigma factor by the core RNA polymerase enzyme. In eukaryotes, the system is much more complicated. The same fundamental ideas and patterns apply. Something binds to the DNA, and then the core promoter goes, or the core polymerase goes on down. There are many, many more factors that compose the RNA polymerase. Things help the RNA polymerase bind. And when I say many more, we're probably talking, there might be as many as 40 proteins to get one gene expressed. It's crazy, crazy complicated. In addition, if we have our DNA in eukaryotes, then we have what we're going to call RNA polymerase bound. There are many other things associated with it, hanging on it, and they're waiting their turn. They're just sitting on it, waiting for a taxi ride down, and eventually they will have their function at some later point in time. We will talk about this when we get into RNA processing, and we get into translation a little bit. Okay? So let me pull up Blackboard here. Or maybe not. So the process of transcription elongation is very, very simple. And we kind of defined this. And we were talking about the abortive transcripts that get produced, or the first nucleotides that get added on. Someone want to be my volunteer? You did it last time, so I volunteer this time. Okay, thanks. You'll be going up and down a little bit. That's fine. Okay, so what happens is basically, and I may need another one. Would you like to help me with this too? So first of all, I want you to watch as I'm doing this. This is actually, come on up, stand up behind me here. So the part of the job during initiation is to separate and create the open complex, and that is about a 10 to 15 nucleotide opening here, right? And it's not easy for me to open this up. As RNA polymerase works its way down, this stuff is all wrapped up, and it's double-stranded. To get access to these nucleotides, RNA polymerase has to melt the DNA, separate the strands of DNA. So it does that, and it works its way down, and now these exposed nucleotides are available. It grabs a nucleotide out of solution, says, do you fit? No. You're the wrong one. Do you fit? No. Eventually it grabs the complementary nucleotide, and it facilitates the 5 to 3 connection. The pyrophosphate goes off the 5' end of the nucleotide and connects onto the 3' end of the nucleotide chain that's been built, right? And so as it works its way down, it continually pushes these helical twists this way. Do you notice something happening here? All of the twists that used to be here have now been pushed this way. So if I had 10 twists in this 100 base pairs, I now have 10 twists in 50 base pairs. Now remember I was talking about, we think for our purposes of just standard DNA, we talked about B form of DNA. And if I crunch the helical twist into a smaller number of nucleotides, we get a different three-dimensional shape of the DNA, and we call that aDNA. What's happening down there? It's loosening. The twists that were here are now slipping down into this area here. So it's not zDNA, which means it's twisted backwards. You can actually untwist it enough that it would be twisted backwards. It's not that case, but it's less than bDNA, right? Okay? So this is a problem. Eventually, I'm going to get to a point where I'm not able to open up the DNA anymore. See how tightly wound that is? Anyone have a solution for this?
Bonds break?
Huh?
Bonds break?
Chemical bonds break? Yeah, that's good for my DNA.
Okay.
Destroy the DNA?
Let it go.
No, continue. I was joking.
I was thinking that it just opens up the strand. That's how it opens it.
Opens up the strand and lets the overwinding happen. Yeah, absolutely. And I will explain the process of that. There's an enzyme called topoisomerase that cuts one of the strands and allows the DNA to relax and then reattaches it. When I explain to you how that happens, you can say, that is utterly stupid. I can't believe my DNA relies on that system. There's another possible solution. Oh, shoot. Because I'm on camera. Ah, man. Now we got to start all over here. Nice. Okay, grab that. I'll send some more down here. Some more helical twists. Okay, I've got a whole bunch of helical twists in here. I want to watch what's going to happen. This is really cool. You probably have seen this and not really understood in this context. I am going to allow a loop to form here. Whoops. Hold that for me. I'm going to allow a loop to form here. And now watch as this thing spins around in here. This is actually a big bubble that forms. If I had more, I would have a big loop down here. And this thing will spin around this way like this. Every time it spins around, it releases one helical twist here. Does that make sense? Well, let's try this again. Let's use a larger piece of film. Send all of these helical twists down here. This thing gets a lot of abuse over the semester here. Right. Let's try this. Hold that for me again. There. Now let's try this. So, as it spins around this way, a helical twist gets absorbed. And that helical twist is now in this twist right here. That is called supercoiled DNA. And it's a way to relieve some helical tension that occurs in these situations. Thank you. That's all I had for now. We'll talk about the importance and the use of this later in the semester. It's my little cliffhanger coming back for next season's exciting episodes. All right, let's try Blackboard here. Lines. Blackboard. Wow, it came right at first sight. Key dog. You guys have probably never seen this end of it, so I'll let you see this side of Blackboard. The dark side of Blackboard. We're doing genetics. Content. Lectures. Same one. Transcription. That's it. That's as far as we've gotten this semester. That's just crazy. Wow. So we've got to initiation and elongation. So, keeping in mind that RNA polymerase, when it opens up another nucleotide and it's trying to map, it grabs nucleotides out. There's a 1 in 4 chance, just a 1 in 4 chance it's going to grab the correct nucleotide to add that will hybridize and be complementary. So 3 out of 4 times it makes a mistake and it keeps having to let go of that and grab a new one. Despite all of those failed efforts, the bacterial RNA polymerase goes at about, if you talk in lab, we say 60 nucleotides a second. Someone is actually calculated. It's 43 nucleotides a second. I think that's incredibly fast. So, elongation occurs by adding nucleotides onto the 3' hydroxyl of a growing RNA. If I got my DNA template down here. So I'll have, this is my 3' OH, this is my phosphate, my phosphate, my phosphate. So this is the 5' end, and this is the 3' end of this guy. So the 5' end connects on the 3' here. So we add from 5' to 3'. We grow from the 5' end to the 3' end. By convention, we write nucleic acid sequences 5 to 3. This is how they chose that. They could have chosen 3 to 5. What does biology do? It starts at the 5 and it adds onto the 3. So we're going to say you write in the order that things get added on. That's why we came up with that convention, okay? It's in red. That doesn't happen often in my class. It's not often I write something in red. Okay. So, what is the effect? I should have left this up here. I had a little bit more to do with this. With my helical coils. So, if the area way down here is supercoiled, you guys can back up real quick. Just do this real quick. So, if this area here is supercoiled, you can see how it gets harder and harder for me to do this. I want you to hold that right there. Okay, now, we've talked about some of these coils from here that slipped into this area. The promoter was right here. Now, let me be another RNA polymerase coming in to make another RNA copy. Twists that used to be here and are now here are getting shoved that way. So you're done transcribing, you let go. Okay, can you hold that for me? twists that were here are now here. Let's transcribe this gene again. Some of the twists that were here are now here and are going to get shoved that way. Every time I make a transcript off of this, I add to the helical twists on this side, and I decrease the helical twists on this side. It gets harder to move this way, as you hopefully can see, right? What's the effect on this side? It's loosening it up. So if you remember, if you can hold that tight again back down there. If you remember, I had a little bit of difficulty separating it the first time. Look at this. Totally ready to go. Once you've transcribed a gene a few times, it's incredibly easy to start again. The DNA just falls apart. When this thing comes in to make the open complex, it's like, oh yeah, totally. Awesome, here we go. Right? So we'll talk about this stuff, especially when we talk about eukaryotic gene expression in WorldSat, in section 3 of the course. Thanks. So these things I'm talking about, they're not just like, oh, cool, supercoiled DNA, awesome. Real functional effects in the cell. So it's actually illustrated here. Upstream, here they have it being negatively supercoiled. That's not true. It becomes less than A DNA, or less than B DNA coiled. And this becomes A DNA coiled, and eventually it becomes supercoiled. And there's a real mess of a problem to solve up there. Any questions on that? Okay, let's talk about termination. Transcription termination. Remember we talked about secondary structures? It's RNA, so it's U, sorry. Okay, I put an RNA sequence up here. Primary structure. This is the 5' end. It has to be, right? Because I wrote it out like this. It has to be 5 to 3. So that's the primary structure. Let's draw a secondary structure for this. See how I did that? Or we call this a stem loop structure. And if I, let's say I could put a bunch of A's in here. And a stem loop structure like that. This stem loop structure is really, really important. And in bacteria, we talk about how transcription terminates. It happens in a row-dependent manner. Rho is the name of a protein. Remember we've got alpha and beta and omega and sigma. This is rho, okay? So termination can happen because a protein called rho comes in and breaks things up and ends it. And then there's a second way that termination can happen in bacteria, and that is rho independent. And what that means is you don't need rho to come in and end it. Let me talk about rho independent first. So as RNA polymerase is running along, and we get the RNA poking out here, the RNA folds into secondary structures. And I want you to imagine here that we get one of these stem loop structures forms here like this. Okay? So these guys are hydrogen bonded to each other. And it turns out this guy can hydrogen bond to this guy. So it kind of pulls this around like that. And then this guy here can hydrogen bond to this guy here. Do I need to draw this bigger? Who wants me to draw this larger? So I can redo it on here. So these guys are all hydrogen bond to each other. And this guy wants to hydrogen bond here, so it pulls that closer. Now those guys are hydrogen bond. It turns out the nucleotide that's here likes to hybridize to that sequence. So we extend this a little further, right? And then this guy likes to hybridize to that guy. So this guy pulls this end a little bit farther in, and they hybridize. And this guy, turns out, wants to hybridize to that guy. Now this is the 3' end right here. This is the OH. And the RNA polymerase is grabbing nucleotides and trying to fit it on there. And what happens is, this guy will kind of tug on it a little bit. The hydrogen bonds are so strong they kind of pull on this a little bit. And they might slip this thing back. So instead of OH being in this spot, it's shifted up this way. And it turns out this nucleotide wants to hybridize to this nucleotide right here. And so the hydrogen bonds pull this even farther out, so that these guys hydrogen bond. Is this making sense? So it essentially zippers itself and just pulls this right out. And RNA polymerase is like, ah, great, I found the nucleotide that hybridizes. Where is my 3' end? I have nothing to react the 5' end of my nucleotide to. The 3' end is gone. In fact, this thing pulls itself all the way out and goes diffusing away. At which point RNA polymerase sits there with a nucleotide. It sits there for a long time, and I think that's like 15 seconds. It's like, this reaction's not happening, sorry. Where's my sigma subunit? Where did my sigma subunit go? And then it goes and tries to start all over again. Does that make sense? I describe it as, or I picture it in my head as a lawnmower that you're pulling the cord on. If you pulled on that and the cord came all the way out, you wouldn't be able to get the lawnmower to go, right? You pull that end all the way out. So termination depends on a specific sequence in the DNA. Now, it doesn't have to be like 10 A's followed by 10 T's. It could be 10 G's followed by 10 C's. You just need that stem loop to be able to form. In the experiments which tested this, if they had a sequence which was 10 G's and 10 C's, if they put a bunch of A's in the G's, you wouldn't be able to hybridize as tightly, could you? And they found that RNA polymerase would not have the nascent messenger RNA pulled out of it. It would just keep on going. It would transcribe way all the way down to the next gene until it hit the termination of the next gene. And then that stem loop structure would terminate it. And you get this huge, enormous RNA produced. Any questions on that? Okay, so that's Rho independent. It doesn't require a protein to come in and do it. Rho dependent, for the purposes of this class, is the same thing, but Rho just binds and helps that process happen. Okay? So we've worked our way through transcription to initiation, elongation, and termination. You guys have any questions on this stuff? Anyone at home? Good, no questions. Good. So here they just show the stem loop structure. Okay, here's a YouTube video you can watch. There's a bunch of really cool YouTube videos up there. And I put a bunch of them on here. A whole bunch of YouTube videos for you guys. Okay, let us open RNA processing. Okay, in bacteria. This is actually today's lecture. In bacteria, we say the messenger RNA is collinear with the DNA. And here were the really interesting experiments they used to run before they could sequence things. So I'm just going to run through it real quick. I don't like teaching history of biology. I want to put this up here so you can kind of understand how it all plays out. If I have my DNA, like this, I'm going to do it in two different colors here. And one of these is the template strand, and one of these is the sense strand, right? So we'll say this is the template strand, and this is the sense strand. It's called sense because it's the same sequence as the RNA that gets made. So let's make an RNA off of this, and that's my RNA. In the old days, even before I was an undergrad, that was a long time ago, they would run these experiments where they would break up the bacterial cells, they would purify the DNA, and then warm the DNA up to a point that the hydrogen bonds that held the anti-parallel strands together, the hydrogen bonds were broken and the strands would separate in solution. That's the template. That's the thing that was copied. And now has a whole bunch of nucleotides that are looking for partners. And here's the sense strand, or the sense strand. And it's also looking for partners. The hybridized too. The needle tube. Complementary bases. So they have all this stuff in the tube. They warm the tube up to about 95 degrees Celsius, not quite boiling. And then they would take it and they would put it on ice to really cool it quick, then set it at room temperature for a while. And what would happen is sometimes this would find its complementary DNA and they would anneal that together, voila, we're back. And sometimes the RNA would get in there first, in which case you would get this. And then eventually, the DNA of the bacteria would come in. And we get this kind of weird structure. Does that make sense? And it turns out, through some interesting tricks, you can actually look at this under a microscope. And on a microscope, double-stranded DNA looks like this. The way they used to compare. If you had an extra piece of RNA in there, then the DNA looked like that. It's a little bit fatter. If you had an area that was single-stranded, or the double strand for some reason couldn't fit, you could actually see the single-stranded part of the DNA in there, if there was no matching DNA strand to line up with it. So you can look under an electron microscope and see it. So if they ran this experiment with bacterial DNA and the bacterial RNA, this is what they got. They get these spots where there's RNA stuck in here. This is a big, huge chromosome. Okay, right. If they did this same experiment with human DNA and messenger RNA that they purified from humans, they got something very different. Let me see if I can remember what that looks like. Yeah. Does this make sense? So what we've got is double-stranded DNA, and then the RNA must be hanging out here. And this is the loop of single-stranded DNA poking up here. We have double-stranded DNA, and then the RNA is stuck in here, and the DNA is hanging around up here. That's been displaced, right? And in reality, it's a little differently. Look like this. Each one of these having a little loop of DNA on it. So we got double-stranded DNA, DNA displaced, double-stranded DNA, DNA displaced, double-stranded DNA. So the RNA must be like this. And the DNA has all these loops around here. So the RNA of one of your and my transcripts is not exactly collinear. There's extra DNA stuff in there. Does that make sense? If my RNA is that, my DNA matches, and then there's like a whole bunch of extra DNA sequence which somehow isn't in my RNA. And then the RNA matches, and then there's a whole bunch of DNA sequence which isn't found in the RNA. So you guys know why. Tell me why. What happens? RNA processing, splicing occurs. What's the alternate explanation for that? It would happen in this stage here. How could you get this result by something strange happening here?
When it was like in solution, and it was trying to find just like a T or like an A to attach, and then that was connected to something else, and it already bonded to it. That doesn't make sense.
No, actually, that's interesting. A T that's already connected to something else, you're building.
Yeah, it was already built, like there was already a strain of something, and it got that solution.
I'd have to think of that in more detail, but there's a pretty simple explanation, which when I say, you're like, oh, yeah, okay, that makes sense. But I'm not going to say it. Anyone home want to call in? Text in to your friend?
So like when the DNA is supercoiled, so it's only able to transcribe those certain portions?
Oh, that's a great one. So if the DNA is getting supercoiled, and those supercoils are going on, so that if the DNA looks like this, we get a little supercoiled area with our loop here, and then it comes back up. What if transcription goes, oh, God, what a mess this is. What a mess this stuff is. I'm just not going to deal with that, and we'll say, like, here's my next nucleotide. Off I go. And they're just covalently connected, and we just don't worry about that stuff. Right? That's a fantastic explanation. That's exactly what they were thinking, is supercoiling happens in specific spots. And this is called dispersive. This is the, I know this is the terms you were thinking in your head. You just didn't say it to the class. Dispersive transcription. And there's this massive search. People would say, there must be specific nucleotide sequences, consensus sequences, located right here. And so located, sorry, right here and right here. On either side of the stuff that's left out. Right here and right here. And those must be highly conserved consensus sequences. And I'm going to be the one who finds it, and then everyone's going to learn the OGG sequence for dispersive transcription. And so the idea that RNA polymerase would be working its way down and then just skip over a little bit of DNA, find some consensus sequence that says, stop adding nucleotides and slide down until you find another start sequence. That was so much easier to envision. The alternative is you make an RNA, and then you somehow covalently, you break the covalent bond here, and you break the covalent bond here, this thing goes away, and then somehow you connect to those covalent bonds. for that to occur, there must be a set of machinery, probably as complicated as RNA polymerase, to do this, splice these things together. And then, what a waste of energy. What if this is 5,000 base pairs here? That's 5,000 ATP that you transcribed. You wasted 5,000 ATP to make that, and then you cut it out and threw it away. Much more sensible. Life has to conserve energy. Much more sensible for there to be consensus sequence. Save that 5,000 ATP of energy and then just start here, and you have your transcript already made. The people who were proposing splicing were damn fool idiots. Who would believe that? When they ran the experiments and they ran the experiments, splicing is it. That's how it happens. The machine that performs splicing is called the spliceosome. You know about the ribosome, the centrosome, and now you know about the spliceosome. Have you ever heard of spliceosome before? Yes, some of you? You've had my classes before or something maybe? Okay, so I'm not going to run through all the details. Here is an electron micrograph image. You can look through this in your own time. It turns out the sequences that are cut out are called introns, and the sequences that are stitched together are called exons. Introns are in your sequence, and they have to be removed. You have to take them out, right? Exons are the things you want. We have now found that there are 3 different ways that splicing happens in eukaryotic cells. And splicing does not happen in bacteria, and I think it doesn't happen in archaea. And I'm forgetting. At what point I am? I think it doesn't happen. So we found 3 different types of splicing, and so we have broken them down, and we've given them names. Type 1 splicing, type 2 splicing, type 3 splicing, and that's it. I think there's actually type 4, which has been recently discovered. And I'm going to show you how type 1 splicing occurs. No machine is necessary. If you take the RNA folds up into a 3-dimensional shape, and the RNA is the machine that cuts itself. That would be me saying, oh, I have appendicitis. Open me up, there's my appendix, there's some infected part of my digestive system, cut that out, throw it away, stitch it back together. Ta-da! Cell surgery. The RNA that's going to be spliced splices itself. Whoa. Crazy. But they're out there. Some people believe that RNA self-splicing is a holdover back to the time when pre-life, when the only things replicating on Earth were nucleic acids and they were not cell-bound yet. That long things of RNA would make themselves and then they would splice into the smaller things which are the actual replicating RNAs. Or you could say, this is so complicated, no way something like that could evolve. Something said that shall be so. I don't know. So let's talk about group 1. So if you take this RNA and you put it in a test tube, a full-length RNA, and put it in a test tube, you will eventually have 2 pieces of RNA. It will splice itself. It does not need anything except a guanosine nucleotide. Okay? So this is what we've got. The RNA, here's my RNA, and here is the guanosine nucleotide. Okay? So that's basically a GTP, an ATP, except it's a G. And I don't think it needs phosphate on it. It's floating around in solution, and the 3-dimensional shape of the RNA folds up so that the guanosine has a binding site. Let's just say the guanosine binds right here. Okay? So there's this is our 3', this is the 5'. We don't normally talk, but this is the 2'. Remember if we were to number, that's the number 1 carbon, number 2, number 3, number 4, number 5 carbon. So This 2' hydroxyl, is it the 2'? No, it's the 3', sorry. 2' is group 2, splicing. So this guanosine is bound in this pocket. It's a puzzle piece that clicks right into the shape of the RNA that folds up. And the 3' hydroxyl engages in a nucleophilic attack. The electrons on the oxygen go after the covalent bond that is the phosphodiester bond that's holding this together right here. And so it attacks this, and it breaks it. So actually, let's do this. It attacks this bond that's of the RNA. So the guanosine is sitting like this, and it attacks this bond here. And it becomes covalently connected. Is that right? Doing that right? No, I did that wrong. It's covalently connected like that. So now, we have 2 pieces of RNA. If you catch this, there's a way they found tricks, geneticists and biochemists have found tricks to freeze the reaction at this point. And they find there's actually 2 RNAs and this guy's stuck on the end. Now, this 3' hydroxyl does a nucleophilic attack over here and breaks that such that these guys are covalently connected. Like this. Such that this thing can go floating away. Does that make sense? Any questions on this? All right, we're going to stop here. We'll get to group 2 and group 3 next time. We'll probably get into capping next time. On Friday. All right. So all the PowerPoint files for this section are online.